Hello friends, welcome to BMH Learning. In this video, I'm going to talk about scar marker, also called sequence characterized amplified region. You must be aware about other random markers like ISSR, RAPD or SCOT. So using these markers, PCR products or amplicons are generated. Now, with the use of scar markers, the PCR products generated from normal markers are actually converted into locus-specific markers. The advantages of doing such manipulation is that locus-specific markers provide sequence specificity, codominance, and high reproducibility. Because of these characteristics, the scar markers are more suitable for gene mapping and varietal authentication. Development of scar markers. So basically, the scar marker generation is a 6-step process. First step is generation of PCR product. Let's suppose we used these markers to generate the amplified PCR product, which is subjected to gel electrophoresis for separation. Let's say this is showing polymorphism in 2 varieties. As you can see, there are many bands, and you are specifically interested in this particular polymorphic band in case of variety 1. Let's assume the size of this band is 600 base pairs. Here comes the main point. This is the DNA fragment, that is 600 base pair DNA stretch, which we want to convert into a scar marker. So what we do? For re-amplification using same RAPD primers. And using 1 to 1.5% agarose gel, the amplicon of interest is separated. Next step is the purification of the fragment of interest. Once the gel electrophoresis is carried out, the band becomes more resolved. The band with the fragment of interest is cut carefully and is treated for gel extraction. There are many kits available for gel extraction. Third step is cloning. So the purified amplicon is ligated into a suitable vector. Once the fragment of interest is cloned, the vector with the interest is transferred into a competent E. coli cell. And to increase the copy number of the vector, the host E. coli is cultured on a suitable medium. As we know, after incubation, we are going to have mixture of self-ligated vector, uncloned vector or vector without insert, and of course, the vector with the insert. So, to identify and separate the cloned vector from the unnecessary stuff, we use blue-white screening method. Fourth step is extraction of plasmid. So the vector is extracted and quantified. And to confirm, we run agarose gel. As we know, we must be having only the cloned vector. We should get only a single band on the gel, describing the size of the cloned gene and the vector together. Next step is sequencing. With the help of standard method of Sanger sequencing and using vector-specific primers, the sequence of the fragment of interest is determined. Final step is scar primer designing. So, once the sequence of the fragment is determined, you design the standard forward and reverse primers. To design these primers, you can use tools like Primer3 or PrimerQuest. Now these primers will specifically amplify only the target polymorphic band. And the thing which we will get here is called scar marker. This was all. Thanks for watching.